Okay, it's a great pleasure to introduce Sarah Perrin Otto, known as Sally Otto. Welcome to everybody. She's going to tell us about the evolutionary enigma of sex. So the evolution of sex and recombination is one of the things that Sally's most known for in her large set of work where she's addressed many, many fundamental problems in evolutionary biology. And Sally is unique in some sense in contributing or combining both theoretical and empirical approaches in her work to address several very, very interesting problems in evolutionary biology. So she's been here teaching in our population genetics and evolution course, and we're very, very excited to hear what she has to say about why sex.
There we go. Hopefully that's better. And thank you to all of the staff that have really made everything go smoothly. I really appreciate it. So I'm going to talk to you today about the evolutionary enigma of sex. What is this enigma? Why is it an enigma? And where are we in understanding this? So across the tree of life, and this is actually across the tree of eukaryotic life, there's an enormous amount of variation in how organisms reproduce. That's illustrated through these colors, where we see everything from the kind of sex chromosomes that determine sex that we have in mammals. Those are the red, and in birds where it's flipped and we have ZW, ZZ systems. But mammals are kind of boring, and if you look across the spectrum of life, you have everything from ploidy determining sex, whether you're haploid or diploid, to having both sexes in the same organism, hermaphroditism, and that's very, very common in flowering plants. In gymnosperms, they do something a little bit different. They have both male and female function on the same plant, but they tend to be in different flowers, monaeae. And rarely you have dioecious and androdioecious and all sorts of other complications in plants. So you see pretty much everything: sex determination, environmental sex determination, no separate sexes at all. And yet, the vast majority of these organisms reproduce sexually at least occasionally. Maybe not every generation, but over a few generations over time. Among flowering plants, about 1% of them can produce asexual seeds, but purely asexual species are super rare and very recent. So they seem to blip out quickly. We don't have very deep lineages of only asexual plants. In animals, very exclusively asexual animals are very rare, less than 0.1%. So the vast majority of animals and plants, over 99.9%, reproduce sexually sometimes. When I first started in evolutionary biology, there was a big mystery about when did sex first arise among eukaryotes. And it was thought, oh, well, if we can figure this out, we can figure out what's different between the asexual lineages of eukaryotes and the sexual lineages of eukaryotes. We can look in and figure out what that transition might have involved. So people have addressed this question in a number of ways, and one of the ways was to look at the genes that are important for meiosis and sexual reproduction. So then I'm showing you a table from a paper from Logsdon and Shurko, and they looked across those genes, and you know what they found? They found that those genes were common to every single eukaryote. They might be missing one or a few, but the core machinery for undergoing meiosis and syngamy was present in everything. So there were no deep asexual lineages that didn't show signs of meiosis. And the conclusion from that is, oh, these things that we thought were these ancient asexual lineages actually aren't ancient. They're up in the crown. One of the most classic examples of this was Microsporidia, which when I was first studying was thought to be one of these asexual early diverging eukaryotic lineages. Turns out it's a fungus, well up in the tree of eukaryotes. And that turned out to be true for many of these oddballs. They were just rapidly evolving, but they weren't deep lineages. So all living eukaryotes, including exclusively asexual lineages, descend from a most recent common ancestor that was already sexual 1 to 1.6 billion years ago. So the evolutionary enigma of sex refers to the fact that sexual reproduction evolved early and has been maintained in the vast majority of species. And it's common, it's prevalent in over 99.9% of eukaryotes engage in sex sometimes, but it's costly. And those costs are substantial, as I'll mention next. What are these costs of sex? There's a bunch of them, and I want to just recap some of the bigger ones. One of them is the cost of producing males. And if we think about a female that's asexual and compare her to a female that's sexual, and just imagine, we'll keep it simple, that she can produce 2 children. If she's asexual, she produces 2 daughters. If she's sexual, she divides up her resources between sons and daughters. I'm keeping it simple, it's half and half. And then those daughters themselves give rise to 2 offspring. But the sexuals are dividing up their resources between sons and daughters, and that population's not growing. By contrast, the asexual population has doubled because every female is making 2 females, and that's making 4 granddaughters, and so on. And it doesn't take too long before this population is going to be overrun by asexuals if they're competing in the same environment. So sexual females, by dividing resources between daughters and sons, should be quickly outcompeted by asexual variants, all else being equal. And even if they're hermaphrodites, the same reasoning holds true. By dividing resources between pollen and eggs, an individual can produce fewer offspring than if she just produced asexual eggs. So that's one enigma, cost of sex. Another has to do with breaking apart gene combinations that work well. Is poker familiar with a card game? Somewhat. I don't actually play it much, but what's a very common Indian card game? Poker. Okay, good. We'll just stick with poker then. All right. So with poker, as with any card game, it's not actually the cards that matter. It's the interactions between the cards that matter. A good hand is because those cards work well together. And the same is true for genomes. That organisms that have survived to reproduce have survived because their genes work together. And just to drive this point home, here is a straight, right? And here is 3 aces. And in poker, those are really good hands. So if you have one of those hands and you just won a bunch of money, and the poker hall says, okay, you've got a choice. You can either keep your hand or mix it up with another hand. Which would you like to do? You'd keep your hand because those cards worked well together and they gave you a lot of money. And so the parents that have survived to reproduce have one thing they know, and that is their genes work well enough to allow them to survive. And if they engage in sex, there is no reason that their offspring will necessarily have a genome that works well in that environment. And this is a kind of profound problem. Sex recombines the genes that in parents who survived to reproduce. They know one thing, they have had enough fitness to produces offspring where there's no guarantees. So that's the destruction of favorable gene combinations. In addition, you have to find a mate, you have to woo that mate, you may fail to woo that mate and remain unmated. There's the risk of disease transmission or predation and all sorts of other things during the lengthy process of mating. For the yeast that we do experimental work, it takes about a week for them to go through the sexual process compared to 90 minutes for the asexual process. So there's lots of costs of sex. What pays those costs? Why is sex so common if it's so costly? In some way or another, most explanations of sex rely on the idea that sex produces variation needed by selection, by natural selection. Indeed, this is one of the first comments about sex and why it has evolved. It's from Weissman at the end of the 1800s, who wrote, the communication of fresh ids. So remember, this is before DNA was known. There was something that was being passed from parents to offspring. Those were ids. To the germplasm implies an augmentation of the variational tendencies, and thus an increase of the power of adaptation. So his view was by combining different traits from the different parents in the offspring, more variation would be produced, and that would facilitate adaptation. So in order, it's kind of it's a nice idea, sex generates variation. And so I have to deconstruct this. And so the first task for me is to prove to you that it's not always the case that sex increases the amount of variation. And the second thing I want to do is show that that variation is not necessarily helpful. It does not necessarily improve the powers of adaptation. So to do this, I'm going to do some cartoon models, just simplified models, but the main principles hold true even for more complex models. So here's a simple model. One gene in a sexual diploid where the gene influences some trait of interest, height. And I've drawn the initial frequencies of those three types in a population at Hardy-Weinberg with fitnesses drawn by this parabola. So the heterozygote's pretty low in fitness, and the two homozygotes have high fitness. Maybe the little ones are too small for a predator to bother one with, and the large ones are just can outrun the predator. So they're fit, but the intermediates aren't. What happens after selection? After selection, this is with the product of those fitnesses times those frequencies shifts the frequency distribution as shown. So what happens with sex acting upon this population? Oh, I'm wondering, this is going to be annoying if this keeps. Let me just see if I can. Second, everybody. I'm just going to try and see if I can. Yeah, I think it's ruined. Okay. Anyway, there's references and things underneath. So afterwards, I can share these slides and you can get the references. But the point here is that if I took this population and it asexually reproduced, I get those parents would breed true. I'd get the exact same distribution among offspring, including these really short and really tall individuals of high fitness. But if I produce if that population reproduces sexually, the variation created by selection is mixed up together and destroyed. This population is less variable in height after sex than it was before sex. Sex did not generate variation in height. It reduced it. So here is just a simple toy example. But if selection has built up variation, it's built up extreme combinations, sex is going to take those and break them apart and create more offspring of intermediate types. So sex need not increase variation. Let's flip this around. What if the fitnesses were the opposite and the heterozygotes were most fit? Well, after selection happens, you have very, very few very few homozygotes, and most of the distribution of surviving individuals are those intermediate types. If that population reproduced asexually, we'd still have a lot of those intermediate types. But if it reproduces sexually, we get more variation. See what that means. Uh-oh. I don't know if I want to leave the meeting or not. Okay, something's good. Okay, so if I reproduce asexually, this maintains. If that population reproduces sexually, yes, there's more variation because it took all of those heterozygotes in the middle, it created some homozygotes out of them, and that population that initially had, if you wouldn't mind, I don't know if you can get rid of that bar at the bottom. That's one way. So yes, sex created variation. But remember, those homozygotes had really low fitness. They're dead. They're the individuals that were eliminated by past selection. And so yes, sex in this case increases the amount of genetic variation, but it's creating the very individuals that were low in fitness, the ones that were weeded out from the parent population by selection. So even in this case where I've constructed it, so variation's missing. There's alleles that are present and variable, but most of the individuals are in the same genotype. And that genetic variation is hidden in that heterozygote. And sex and segregation can untie those allelic combinations and produce more genetic variation. But doing so reduces fitness. And if you generalize this, not just at one loci, but at multiple loci, you find that this is a general problem. Whatever gene combinations have built in the past by selection tend to be undone by sex and recombination. That's called a segregation load if it's at each locus. The one we were just looking at was a segregation load because we're segregating these out and making dead kids. And it's called the recombination load when loci at different loci are recombined. But in either case, you can show that almost all of the time there's a major reduction in fitness due to sex among the offspring. And this is a poker problem. Taking hands that have survived, taking genomes that have survived, and recombining them almost always reduces the mean fitness of the offspring. Okay, so I hope that those toy examples are enough to show you that there's indeed, it's a trickier issue than you thought about. Sex doesn't always increase variation. If selection has already increased variation, sex can destroy that. And even if it does produce more variation, often that will be variation that is deadly. So to solve or address this evolutionary enigma, we have to kind of flip this around and say, okay, what conditions are necessary for the variation to be missing, but sex can regenerate? And secondly, we have to ask, would genes that promote sex and promote recombination even spread within a population by so doing? So those are two separate questions that modelers have been trying to address over the last century. What conditions mean that variation is missing from a population, and then showing under what conditions sex and recombination are favored. Okay, so this is going to involve a kind of somewhat technical player or actor in this show that I'm presenting, and that is linkage disequilibrium. Linkage disequilibrium measures associations between loci. In that hand where we had triple aces, there were associations. If I had an ace in one card, the other winning cards were more likely to be aces. So that's a linkage disequilibrium, an association. Some of the associations are good for individuals. They have good alleles with good genetic backgrounds, good alleles at other ones. These represent chromosomes. 2 loci under selection, and the happy faces are the alleles that increase fitness. Selection will then generate, and if the individuals that have really, really high fitness are these individuals and these individuals, for example, then we'll have more of them, and that's a positive disequilibrium. On the other hand, maybe we have a lot of these ones where there's a good allele here and a good allele there, but they're both on genetic backgrounds with bad alleles. So in this first case that I was talking about, there's already a really high variation in fitness. We have extreme genotypes present, the ones that are good alleles at all loci and the ones that are bad. There's already as much maximal differences between the population. But in this case, there's almost all of the good alleles are tied up in genetic backgrounds with bad alleles. They may be similar in fitness, but there's not much distinguishing them. So I've basically reframed the question to say, variation will be missing if it's the case that evolutionary processes in the past have built up an excess of good genes on bad genetic backgrounds. So when does that happen? When do we get predominantly negative disequilibrium? And for the class, you've seen these equations this morning. This is for 2 loci. If there's no disequilibrium, then recombination won't matter. If there's no genetic association, sex won't matter between loci. And that's a little bit like saying if you've already got a well-shuffled card deck, it doesn't matter if you shuffle it anymore. So in order for shuffling to matter, in order for recombination and sex to matter, you have to have correlations in state between the genes carried in a genome and different genomes in an individual. Only then will mixing actually matter. So if we're trying to understand the evolution of sex and recombination between genes, we better understand why are these associations present. Okay. So that's one task that we have to do, is when do we have that kind of association build up? And then the second thing I said is we also have to ask, when would genes increasing the amount of recombination or increasing the amount of sex spread within a population? And this is an approach, it's called a modifier approach. It's a kind of a core area in evolutionary biology, but it's not really well recognized outside. Basically, modifier models ask, how does the organism change the way it lives and reproduces? If it's asexual, under what conditions will more sex spread? If it's hermaphroditic, under what conditions will separation of sexes spread? So those are modifiers that are changing the stage upon which evolution acts. So that stage is constructed with the attributes of a population, whether it's sexual or asexual, diploid or haploid, separate sexes or not. But that stage is itself a product of past evolution. So to understand how it is evolved, we have to ask these kind of questions. When do genes promoting sex spread? When do genes promoting recombination spread? And these modifiers, it sounds like a really obscure thing, but they're the genes that we know, right? They're the things like the inducers of meiosis. So just for example, in yeast, I've got a list of them. IMC1, inducer of meiosis, whether or not meiosis is likely to occur, or mitosis. MFA1, that's one of the mating factor pheromones in determining whether or not sex can happen, because they can identify and mate with other individuals. Recombination is the product of a bunch of different genes. PDRM9 is a known inducer of double-strand breaks that initiates the recombination process. But in addition, SPO11 is really essential for bringing the chromosomes together. REC8 and others are essential for recombination. So all of those genes are the product of past evolution that has favored sex and has favored recombination. So by sex, I mean the coming together of gametes and the separating through segregation. And by recombination, I mean this process where chromosomes are broken and recombined with one another. And we really want to understand both, because both contribute to the amount of genetic mixing of the genomes. Okay, so just to recap, if I'm interested in how much sex a species engages in, I imagine that there is a gene that controls whether or not that yeast cell, for example, reproduces through fission, asexually, or undergoes meiosis and makes a tetrad. If that gene promotes sex, then this is going to happen more often. When will that spread? Or if I'm looking at the evolution of recombination, I could imagine a gene that increases the number of double-strand breaks and promotes more recombination. When will that spread? So just to give you a flavor of the models, this is the simplest model that has a modifier model locus that changes the amount of sex. So that's what the M locus does. And the A locus, A gene, is just responding to selection. So even there, we have a whole bunch. We have 4 possible types of diploid parents, 4 possible types of big M could be with big A, big M could be with little M with big A, little m with little a, 4 chromosomes, and these are combined into 16 parents. Some of those are redundant, they're the same, just where you got your genome from your mom or your dad, but still, that's 10 equations. They sum to 1, so there's 9 equations that are independent. But still, they're nonlinear, and they're messy. So what do we do with these models? We track those chromosomes as they go through selection, and they go through either sex or asexual reproduction. We track which of the genotypes they carry, and so how much sex they undergo. And then we use math to say, I don't want to just know what happens in the next generation. I want to know what happens over evolutionary time. So we do this again and again and again, and use asymptotic dynamics to figure out what will happen. Okay. So I'm going to report on summarize the results of those models. So first of all, are there forms of selection that actually lead to populations that are depauperate for variation? And the answer is yes, if there's what's called negative epistasis. Negative epistasis means that the fitness surfaces are curved downwards. That's the negative part. Things that are all capital letters or all lowercase letters are on average less fit than the intermediate types. So selection is acting kind of on average against the extremes and on average for the intermediate types. And what happens over time is that we're left with a lot of intermediate genotypes and we lose the extreme genotypes. Here's an example, fitness. Instead of my arms, here are fitness functions that I'm drawing, where you're looking at either having 1 or 2 beneficial mutations. And so fitness rises with more mutations. If there's no if the fitness effects are independent, that's multiplicative interactions. And the negative ones are less show more negative curvature than that. And so in this case, the average fitness of the extreme genotypes, on the blue with negative epistasis, is lower than with 1 on a log scale. And that's true whether we're talking about beneficial mutations or deleterious mutations. Whenever there's this negative curvature, we can show that negative genetic associations develop. That what comes over evolutionary time with those type of fitness surfaces are an overabundance of genotypes stuck in the middle without a lot of variation. So to recap, with negative epistasis, extreme genotypes would be less fit on average. They then become less frequent after selection because on average, selection for those extreme types is not promoting their frequency as much. And that decreases genetic variance in fitness. That decreased genetic variance in fitness isn't because the alleles are gone, they're still present, but they're tied up with good alleles in bad genetic backgrounds. This measure of negative linkage disequilibrium that I've talked about. So what about a modifier of sex or recombination? So sex, if that's what's been happening, if there is a preponderance of those type of fitness interactions, and we've got an abundance of these good alleles tied up in bad genetic backgrounds, then sex can increase the genetic variation and release those alleles from their bad backgrounds. Sounds great. Sex could restore genetic variance and improve the response to selection. Models that look at modifiers and do they then spread have found, though, that epistasis, it's not enough for epistasis to be negative. Negative epistasis will generate the right type of negative linkage disequilibrium and associations, but when sex happens, it also makes those really unfit kids. And the net result of that actually selects more strongly against the modifier, despite the fact that it increases variation. So that's a summary of the modeling results, but these modifiers that increase sex and recombination, even though they produce more variation, the fact that they are make unfit kids selects against them. And so only if that epistasis is negative and weak is the sum total positive and does the modifier spread, more sex spread in the population. Maybe even more fatal than that is there's this teeny little zone in which sex can be favored. It has to be negative and not too negative. But if there's variation, some loci have positive interactions, others really strongly negative interactions. There's a lot of, we know that's true, that different pairs of genes interact to determine fitness in a wide variety of ways. But that variation is fatal because most of the time, positive epistasis always selects against sex. Strongly negative one always selects against sex. And so even if on average we're in the right teeny little zone, accounting for the variance, it's not. That teeny little zone does expand if there's hardly any sex and recombination in the population, but if we're trying to understand sex and recombination in eukaryotes, it's not that rare. So this is kind of where we were at. The model said that that yes, sex could spread, but it's unlikely it can work, but an epistasis not only has to be negative, it has to be negative and weak. There can't be much, and there's no empirical data for all of those three things being simultaneously met. So for something as common as sex, this seemed like really shaky ground that we were on.
Sorry, can I ask something? Yeah. You mean by the second point, variance over different genome types, right?
Different pairs of loci.
Pairs of
yeah. So it's not just A and B, right? But it's C and D and C and E and all those pairs.
What about something like variance over time? Like, for instance, selection rules will vary over time, right? Over evolutionary time.
Let's go back to that point, because that's kind of a different explanation for sex. This is the negative. There's something about the world that the fitness surfaces make variation missing, that sex and recombination can spread. That's another alternative, which is called the Red Queen hypothesis, that the world changes fast enough. But let's come back to that one. So this is our evolutionary biologist spherical round cow. Basically, I would like to argue to you that we have been modeling infinite deterministic populations, and that's like modeling spherical cows in evolution. And that we were actually missing the core elements of why sex was even favored by this cow. What was missing? What was that spherical cow? I would argue that it's the finite nature of the genome combinations in a population. This was first noticed by Felsenstein in 1974. And he noticed that there was a pattern emerged when comparing those papers which found an advantage to recombination and sex between non-interacting loci and those which did not. So he subdivided the literature, and he noticed that the ones that treated the populations as infinitely large using deterministic tools, like the negative epistasis hypothesis I was just telling you about, didn't actually find an advantage to sex. And the ones that were finding consistently an advantage to modifiers or an advantage to having more sex were those that were treating the finite nature of populations. And that's weird for us to think about, because most of the time when we think about drift and stochasticity, it just creates a little bit of wobble in our evolutionary trajectory. This is saying something different. This is saying if you don't include that little bit of drift and that stochasticity, you actually get a different answer. Rather than having sex favored, you have sex disfavored. Okay. So here's my claim, that the advantages of sex come from the challenges of the finite options in any real population, of combining the many, many, many different types of possible alleles that are present in a whole genome. It's a combinatoric problem. And this is worse after a period of selection, because after a period of selection, the best chromosomes, the ones that work well in that environment, are the ones that are remaining. But there's going to be very little fitness variation among them. And this problem is a combinatoric problem because there's billions of base pairs in our genome. So there's lots of possible combinations of chromosomes, way more than the population size of any species. Okay, so with all of those combinations, any species has only a limited number to begin with, because it's got a population size way less than 4 to the number of polymorphic loci. And then after a period of selection that winnows out the worst of those, it's got an even more restricted subset. And so, but without accounting for that stochasticity, we're not going to capture the advantages of sex. So putting on these happy faces and sad faces to represent what we might see across the genome is we might see some nucleotides carried by an individual really hurt, some are kind of mild, some kind of mildly helpful, some really good, but selection doesn't act on those individually, right? It acts on the combination. The alleles that you all carry all throughout your genome. And so selection can't act on any one of these separately from the others. That's this problem of selection acting not just on one allele, but on the sum total of its genetic background and that allele. This idea actually, the kind of core of it, the clues of it, goes back a century to something that Fisher wrote in his book. He said, so the R.A. Fisher and H.J. Muller were early evolutionary biologists, population geneticists, and Fisher wrote, unless advantageous mutations occur so seldom that each has had time to become predominant before the next one appears, then they can only come to be simultaneously in the same gamete by means of recombination. So he's saying, we've got a problem here. Without sex, you might have a beneficial mutation, you might have a beneficial mutation, but they're separate, in separate individuals that can only come to be together through sex and recombination. He didn't say anything about stochasticity or infinite population sizes or anything, but had the population been infinite, then that beneficial combination would have been instantaneously produced by mutation. So this is a problem of a finite population. Muller argued a similar thing and said that without sexual reproduction, the various favorable mutations that occur must simply compete with each other. They can't come to be together without sex. This has been drawn in a really classic way of illustrating this problem known as Muller diagrams. This is from a paper in Crow and Kimura. Time is along the x-axis, and we're representing the variation within a population along the y-axis. If the population, and these are all beneficial mutations that arise and spread, but here is an example where 3 different beneficial mutations happen in different individuals, and with no sex and no recombination, they compete. Only one of them eventually makes it. After a period of time, a second beneficial mutation may arise in that genetic background, but they have to be nested in this way. Whereas in a sexual population, this individual, one of these individuals, could mate with one of these individuals and produce the favorable combination of the two. So Crow and Kimura in this paper were asking how long a delay does that cause, and they pointed out that it can be a really large delay. That the waiting time from the first to the second spreading beneficial mutation, given that they had to be nested like this, was much longer than if they could be put together in a sexual population. And again, this relied on there being a finite number. It's not a deterministic problem. And this was one of the papers that Felsenstein was summarizing. So just to recap, they showed mathematically that sexually recombining populations could adapt faster, much faster in large populations with many weakly beneficial mutations. And I mentioned that. A second paper that was really essential to these arguments was produced by Bill Hill and Alan Robertson in 1966. They were quantitative geneticists, and they were really trying to understand what are the limits to breeding programs, domesticated breeding programs, trying to improve cattle stock. It was a big issue that Bill Hill's been involved with. And he wanted to know what are the determinants of how fast evolution by natural selection could occur. So they did some early simulations and showed that even though populations initially had no associations between genes, they were initially in linkage equilibrium, no correlations, the advance under selection can be reduced when genes are tightly linked, even if there's no fitness interactions between those genes. This was kind of a surprise. This took the literature by surprise. In short, selection at one locus always interfered, or sometimes it was negligible, with selection at other loci, reducing the fixation probability or the spread of beneficial alleles, even if they weren't interacting. What's going on? Well, to answer that, we have to be able to tackle these stochastic models. And as I said before, the dynamics are not great. You know, even in the simplest case, we were dealing with 9 nonlinear equations. More loci, it expands. And now I've got to add drift to this. But we have to be able to tackle that if we're going to answer this question. One of the first kind of attempts at some insight came from Hill and Robertson's paper. And they pointed out that this linkage disequilibrium, remember that's the statistical correlation between alleles carried by different individuals. And they pointed out it has this relationship to the disequilibrium in the previous generation, where these are just some constants that depend on the recombination rate and the selection. But they're positive. So one point about that is if there's no genetic associations, no correlations in one generation, there won't be in the next generation. But what if there's variation? What if there is stochasticity and drift around this? Then by chance, sometimes there's positive covariance and sometimes negative. But if you calculate the expected value of the disequilibrium over that variance, it's negative. So here's just an illustration of that equation with the expected value in the next generation. And And there's no associations. These genes aren't interacting with one another. But on average, in the next generation, that disequilibrium goes down. And it's related, I don't know if I have it in the next slide. Yeah. It's related to this argument that I've made about good genes and bad genetic backgrounds. When by chance a positive disequilibrium develops, good alleles with good, they really rapidly rise in frequency, and the bad ones go away, and that disappears quickly. When by chance there's the drift, the chance events, couple good alleles on bad genetic background, that sticks around. And the average of all of that ends up with these kind of chromosomes stuck in this intermediate zone. And all that requires is considering the finite nature of the world. What Hill and Robertson also pointed out was, given the complexity of the simulations, they concluded that it seemed unlikely that any general theory could be constructed. And probably this is where my naivete as a graduate student should have led me to stop. But it was like so important that this field go in this direction that we did construct those models. And one of my postdoctoral supervisor and colleague, Nick Barden, and I set out to tackle this problem in a number of ways. In the 1995 paper, what we basically asked was, we used a multi-type branching process to say, would a modifier, by uncoupling alleles from the genetic background, improve the fixation probability of beneficial alleles so much that it rises in frequency with those combinations? And so that's one way we were able to handle the stochasticity. The other approach we've taken is to say, maybe we pay attention to the wobble caused by drift and stochasticity, but we keep track of the means and the variance of that wobble and the associations they create. And so that was these methods to track moments. But now we're tracking moments, not just in the mean, but the variance and covariance between all of these chromosome frequencies. And we were able to show, here's an example from that tracking of moments example. We were able to prove that a modifier increasing the amount of sex and recombination would spread, alleles promoting that amount of sex would spread, even though in an infinite population they wouldn't. And we were able to confirm the predictions with simulation. I would have to say, though, we had to make a number of assumptions along the way. And this is an area, multi-locus stochastic modeling, that continues to be a really core area where more mathematical approaches would be useful. We were able to show with a similar analytical method that selective interference is important even if the populations are really large, and that was surprising, especially if selection was weak, more loci, or spatial structure. Here's an example of what I mean by working even in large populations. This is a simulation which looked at how much more sex is there following a period of adaptation at 2 loci. So we're tracking these modifiers and seeing if they spread and if more sex is occurring. This is the population size. And the different lines are different forms of fitness interactions. Remember, in an infinite population, I said you had to have negative epistasis? And I've chosen those ones. So this is an infinite population. We only see more sex if the epistasis is negative, and all of those are weak from this in this simulation. There's positive epistasis, we don't see anything. But in finite populations, that's almost irrelevant. The fitness surface is hardly a driver of the advantages of sex. Indeed, we find even more sex being favored if there's positive epistasis. What really is a driver are these chance associations where the population, after a period of time, runs out of fuel. And that fuel being differences among the different chromosomes. There's still genetic variation within them, but they have to be untied. So this works across a range of population sizes, including a range of population sizes, a range of epistasis, and we want that. We want a mechanism that's robust, that doesn't depend so sensitively on being in just this kind of fitness interaction in order to explain something as common as sex. And I have to say, I was expecting it to work well in small populations, and over time I've come to realize that actually that's not such a constraint. And even if the population's infinitely large, selection is not experienced at that infinite scale, it's experienced locally. And these stochastic processes are happening locally. So we were able to show that even if the population's infinitely large, but the disequilibrium is arising by chance within each spatial location, it still works. The other thing that happens as you go to larger and larger populations, it actually maintains more variation. So the problem of interference is actually worse in a large population because there's more sites at which you have polymorphism, genetic variation. So it gets more and more sluggish. Selection interferes more and more as the population size gets larger. And you don't need to just consider a positive selection. It works with deleterious mutations too. This is just looking at a genome with a deleterious mutation rate and asking how much, these are modifiers that arise one after another, and we asked how high would the map length evolve to in this simulation. And it turns out that any form of selection can work. It can be deleterious mutations, beneficial mutations. It can be these host parasite cycles because they're generating, as one favorable allele spreads in the host, it's causing selective interference with all the other loci in the genome. And any combination of these selective forces. And I didn't put in the summary slides, but it turned out that the red queen by itself in an infinite deterministic model, it doesn't work very well. And the reason for that is the same argument, that the alleles, that the individuals that survive in the population today are the ones that have recently been able to survive the parasite attacks. So most of the time, recombination and sex are breaking apart the very combinations that allowed their parents to evade the parasite. The only way it works is if you have such strong selection that there are tightly coupled systems with gene combinations that are favored now being actually killing you in just a few generations, which we don't think is very, very common. But this is probably very common, that it contributes, that those type of interactions between species contributes to the density of selection witnessed across the genome. And so the key element, I would argue, is selective interference, that there's just enough sites under selection across the genome to favor modifiers that maintain sex and recombination or increase it if it's absent. I put this slide on, and just in case you're a population geneticist and you've heard these other terms, that this selective interference is actually an umbrella term that encompasses all of these explanations. Clonal interference, Fisher-Muller, Muller's draft, Ratchet, genetic draft, Hill-Robertson effects, they all are talking about selective interference causing genes at one locus to not be independent of the selective response at neighboring loci. We can talk more about that if you wish. So just quickly to wrap up, what's the evidence for selective interference? So I do think that since the start of my career to now, that this that we've got a much more solid theoretical basis to say, yes, this will work. It's pretty robust. And increasingly, we have more data about whether or not selective interference is a big deal or a small deal. So the first type of data that we had was comparing how quickly sexual populations would adapt versus asexual populations. And there's a large number of studies that have looked at that and found that sexual populations adapt faster. This doesn't prove the mechanism, though. It could be due to selective interference that unties beneficial alleles in one bacteria and puts it together with a beneficial allele in another bacteria, and that allows it to respond faster. Could be, but maybe the fitness surfaces were those negative nymphostatic ones. Or maybe the genes that promoted sex actually had other beneficial side effects. So we can't tell, not from that experiment. I think the best picture of selective interference comes from actually genomically sequencing populations as they adapted over time. So this is a study of yeast from Mike McDonald. And at the end, they figured out which mutations that occurred were beneficial in gold and which ones were deleterious in blue. And then they got their stocks out of the freezer and figured out, well, what were their frequencies in the past? So they reconstructed this process. And the top graph is for an asexual population. What you can see are these kind of periods where something is sweeping through and it's dragging a bunch of junk with it too. So this one, something is rising in frequency, but so too are these deleterious alleles in blue. So it's only lucky cohorts that seem to establish. Whereas in a sexual population, they're not fully sexual, it's just once every week they go through sex. Then you see something very different. Beneficial mutations are rising and fixing kind of more independently of one another, not just in those cohorts. So beneficial mutations were more likely to fix, more of them did fix. And if you look at those deleterious mutations, they never saw those deleterious mutations hitchhike so high that they fixed in the sexual population. But many of them did fix in the asexual population because they were stuck in the same genome. Those unhappy faces were stuck in the same genome with the happy faces that were spreading. So this is a picture of selective interference. It's exactly what the model would predict. A synthesis of field studies looking at whether sexuals adapt faster than asexual populations finds that in most cases there's support for the mechanism that asexuals do accumulate deleterious mutations more rapidly than sexual lineages. They concluded there was not enough evidence for exactly how fast asexuals and sexuals adapt, but they could look for deleterious mutations. So this was consistent as well. Again, here we don't know the mechanism, we just know this comparison. And people are looking inside recombining genomes and finding that in regions where there's less recombination, there's more signatures of selective interference. So that was measured by these runs of homozygosity, and those were stronger in regions of low recombination. And finally, people are starting, Anil Agarwal and those Becks have been looking at experimentally testing these by using rotifers that can produce asexual offspring or sexual offspring. And then they test and see under what circumstances does the frequency of sex increase. And so I think more such experiments are needed, but in their experiments, what they were finding was in the adapting populations, the frequency of sex stayed high, whereas in the control populations, it went down. And it was consistent with selective interference, but again, it's hard to get at the mechanism. Whereas I think this picture, we can see the mechanism because we have these correlated frequency fixation events, which is saying that the deleterious mutations are not uncoupling from the beneficial ones. So I think that's where we are in this literature. Selective interference, it's an umbrella term that refers to the fact that selection doesn't act on individual genes in isolation, it acts on genomes. And those genomes have so many possible combinations that are not experiencing selection because they're tied together, unless they recombine and engage in sex. So selection in the presence of drift causes the accumulation of go alleles on bad backgrounds. That's what's going to persist over time. That limits the response to selection. Sex and recombination can evolve and be maintained due to selective interference. And so is the evolutionary enigma of sex solved? I think so. I think that this problem, this combinatoric problem that evolution faces is a huge one. There's just too many sites in the genome, too many possible combinations. I think selection is acting at enough of them that this interference is a big problem. But I think empirically, we still need to nail this down. We still need to have more examples like the McDonald one that is really showing that actually, yes, selective interference is strong. And we need to know more about the density of selection acting across the genome. How often are there sites that go from low frequency to high? How often are there sites that are fluctuating elements due to selection with interacting species? What's the density of deleterious mutations? What's the density of sites that are experiencing selection, even if they're silent substitutions? So I think all of that together will help us understand if indeed the density of selection across the genome is strong enough to explain the evolution and maintenance of sex in eukaryotes. And with that, I would like to thank my many co-authors and thank you.
So where you talked about that in finite populations, variation would be coming down, and the only way beneficial mutations could come together and not compete is by sex.
Say that again, that the beneficial mutations could
Beneficial mutations could come together by recombination,
Yes.
and that's happening during sex. So I was wondering, like, if a mutation has a significantly large pleiotropy, like the degree of globalness of that mutation, and if it's like globally beneficial,
Yep.
so can that be a loophole to not mutations coming together, like beneficial? Yes, and that can help with asexual reproduction.
Yeah. So I've kind of tried to keep things simple by just labeling things as beneficial or deleterious, but the math itself pays attention to how beneficial and how deleterious. So if you have a strongly selected site that's really beneficial across all possible environments, it sweeps really high to fixation. And we've been able to prove that it can drag along with it deleterious things nearby that are of smaller effect. So the strongest effect nearby are those really strongly beneficial mutations. But on the other hand, that doesn't last very long because it rises and fixes. So yeah, the net effect of a bunch of small effect mutations.
The bad background thing.
What was that?
The bad background.
The bad background can fix along with a good background. Yeah.
And yeah, I didn't quite understand that how both positive and negative epistasis can lead to selective interference, and like how abundant positive and negative epistasis are in the living world.
Right.
As such.
So that's a very good question, and it didn't go into it. But the point is that with drift and selection, after a period of evolution, you end up with an abundance of good alleles on bad genetic background. Now, that's like all of the bad poker hands. You recombine them, and actually you can increase fitness. And that increase in fitness is even higher if the good combinations are really good, like the triple aces. So basically, I've reversed the poker game problem. Evolution in finite populations tends to accumulate not great hands, and sex and recombination can improve fitness. So it doesn't actually, the recombination load, when there's positive epistasis and selective interference, turns into a recombination benefit. I didn't get into that, but that's what the math says.
Thank you.
Hi. So does this theory of sexual interference, does this give any new explanations for those blips of asexuality that evolve?
Yeah.
That's my question.
Yeah. So when do we ever see asexuality? So very good question. If the environment is stable, then we'd expect to see asexual reproduction happening more often. If the costs of sex are really high, then we'd expect to see asexual reproduction more common. And I think that's probably the more proximate driver. When are mates rare? So we see these correlations that asexuality tends to be more common in northern latitudes or nearer the poles. And I think that's in part because it's harder to find a mate. If the density is really low, then the costs of sex become higher.
Thanks.
Hi, the questions on your previous slide, your summary slide, I guess. Is there an empirical approach to understanding this? Because we do have track of a lot of species, right? In some sense, I'm not sure how good of a track it is, but to understanding why sex is so common in some species and not. Like in your first slide, also you had this tabulation of all these species, camels and whatnot.
Right.
So
I think science works by kind of eating at the edges of all of these questions in multiple directions. And I think that's what we're doing as a field. So many talks in the last week have talked about estimating selection and where it's acting in the genome. And we're accumulating evidence of just how much of the genome is under selection. But even then we can miss a lot, right? Because there can be just really short periods of time when something is favored, or maybe in the winter something is favored and in the summer it's disfavored. But those kinds of things are also being studied. So I do think we're getting a better handle on what's under selection across the genome. And as Deepa mentioned, the things that we thought were neutral often aren't. But still, no, we have no species for which we have an accurate measure of the density of selection acting across the genome. And then the flip side of that was I have glossed over how much sex should each species have. And it is true that most of these advantages come from having a little bit of mixing. A little bit of sex goes a long way for bringing together alleles from different backgrounds, from separating them out. But if the density of those selective sites is high enough, you need a lot of mixing. So I think we don't know, are mammals and birds obligately sexual because of so much selection happening in our genome? Or is it another reason that might be boring? Like, it might actually be hard to maintain the ability to have sex and to be able to reproduce asexually. And so maybe it's a more boring reason that we've accidentally lost the ability to have asexual reproduction, or some combination. So I think that when I say, do I think the evolutionary enigma is solved? I do think that this is the core of the explanation, but there's still a lot of questions around that.
I see, thanks.
Yeah.
So let's thank Sally once again for a wonderful lecture. As a token of appreciation, ICTS gives you this.
Oh, I thank you.
Thanks to Sally also for a whole week of lectures.